Hi guys, so what I'm going to be talking about today is a study done in 2019 about dopamine neurons in the ventral tegmental area. So what are dopamine neurons and what is the ventral tegmental area? Dopamine neurons are brain cells that are involved in reward processing. So whenever you do something that's rewarded, like say you're playing a video game and you do something that wins you the game, your dopamine neurons will be activated. The ventral tegmental area is in the midbrain, it's this blue dot right there, and it is highly involved in the development and expression of behaviors associated with drug use. So there's a lot of dopamine neurons in the ventral tegmental area, which I'll just call the VTA from now on. And in this study, these researchers wanted to figure out if these dopamine neurons have the same function, reward processing, or if they're multiplex, meaning they have many different functions. So in order to figure this out, they took a bunch of mice and set them up in a virtual reality maze. So they would cue the mice to go either left or right, and if the mouse turned the correct direction, they would be given a reward of water, and if they turned the wrong direction, they would be given a tone and a timeout. So in order to actually see what these neurons were doing, the researchers used a molecule G-CAMP, which they injected into the mouse's brain above the VTA, and a 2-photon microscope, which is just a special optical technique that allowed them to view deeper than the surface level of the brain. So the molecule G-CAMP glows whenever there's neuronal activity, meaning that when one of these dopamine neurons was activated, the researchers were able to see that as a change in the fluorescence of this molecule. And using this technology, they were able to see, based on what the mouse was doing, which neurons were activated when and for what variable. So they found that during the cue period of the maze, which is when they cued the mouse to go either left or right, these dopamine neurons were multiplex, meaning they were active for many different variables. But during the outcome period, which is when they were either rewarded or punished based on their decision, they were all pretty much focused on reward processing. So they took the 8 variables that they saw these dopamine neurons were active for and made graphs for their change in fluorescence, the neurons' change in fluorescence. So there's position, which is the mouse's position in the maze, speed, which is the mouse's speed throughout the maze, acceleration through the maze, etc. So we're just going to look at this graph in a little more detail. So this colored graph, each horizontal line represents one neuron. So as you can see, the colors fluctuate throughout the graph, meaning that each different neuron has a different response for different positions in the maze. So the researchers took out one of these neurons and made its own separate graph to kind of break it down a little. So the x-axis is the mouse's position in the maze, and the y-axis is the neuron's change in fluorescence, meaning its change in activity. So as you can see, as the mouse progressed further through the maze, the neuron increased in activity. So this is just an example of how these dopamine neurons in the VTA are not only multiplex, but within these various functions, each neuron behaves uniquely and differently from one another. So that's all I have for you guys today. The link to the article for this study is in the description below. Thank you guys for watching.